Hello, good afternoon, class. Let's get started.
Hello.
We will be doing lecture 4 today, which is about membrane transport. So basically things getting in and out of the cell. And I think we're gonna have a little bit of time at the end to talk about your lab as well, which is going to take place next week. So when more people come in, we'll talk about that. But first, let's go through the membrane transport um, study guide here. Actually, you know what? Before we fill this in, I think it's probably a good idea to draw the cell membrane just to refresh your memory about what's the major components are and the function that they serve in the cell membrane. So we have something called a fluid mosaic model. Okay, so the fluid mosaic model is actually I have the same thing here. Okay, so the fluid mosaic model, basically shows the various component of the cell membrane, and it's fluid because it's not a rigid structure. Okay, it actually flows around, and it has various component to it. That's why it's called mosaic. So why don't we start by drawing a cell here, and there is your nucleus, and we're gonna take a slice of the cell membrane, and we will zoom in. Okay, we'll zoom in on the cell membrane and see what it is made up of. So the cell membrane is going to have these phospholipids as the major component. Okay, so some phospholipids will have unsaturated fatty acids. Some of them will have saturated fatty acids. That's what these kinks are, right? So there we go. You should probably draw along with me. So that's our hydrophilic head. And this would be the outside of the cell. Okay, outside. And this would be the inside of the cell, the cytoplasm. So the hydrophilic head either faces the outside of the cell, which is mostly water, or it's going to be facing inward in the cytoplasm, which also is mostly water. Now, in between the hydrophilic heads, you would have the hydrophobic tails. Right, so it forms a nice barrier that helps block unwanted trafficking across the cell membrane. Now, every now and then, you might have a protein that goes all the way through your cell membrane. So I'm just going to draw more phospholipids here. Okay, and you can just randomly put some unsaturated ones every now and then. And the protein, if it goes all the way through from one end to the other, this is what we call an integral protein. Integral protein. But the protein doesn't have to go all the way through. They can just be attaching to one side. And in that case, we call it a peripheral protein. Okay, so here's another one. Think about peripheral protein like toppings on a pizza. They just, lie on the top, doesn't go all the way through, right? Integral protein is more like raisin in a raisin bread. You can see the whole thing from both sides. So the phospholipids, right? They are able to move side to side, move horizontally, like this one might actually just swim around to the side, and rarely, very rarely, will they flip to the other side. Yeah, sometimes they do flip, but it's a rare occurrences. So these phospholipids can come in two forms, right? So these are saturated phospholipid. And then these ones with the kink, that's the unsaturated. Now, having more saturated phospholipid is going to make your membrane less fluid. On the other hand, if you have more unsaturated phospholipid, it will make your membrane more fluid. Okay. So it's not like one is better than the other. It depends on the environment that the cell is in, right? And we're going to talk more about it in a bit. Now, the in addition to the phospholipid, you will also have some cholesterol. Right? I'm just going to represent the cholesterol with these 4 rings. Right? So that's cholesterol. And the function of cholesterol is to help modulate, helps control or regulate membrane fluidity. Okay, you don't want the membrane to be too leaky, too fluid, because everything would just go in and out without much regulation. You don't want it to be too solid either, because then nothing can come in, nothing can go out, right? So by having the right amount of cholesterol, you can help control the fluidity. Of course, the amount of saturated and unsaturated fat will also help control the fluidity. So generally speaking, more cholesterol equals to more fluid. Okay. Now I'm just gonna add a few more phospholipid here because I'm running out of space to draw more things. Now this thing, of course, it's like a whole sheet. It's more like a mattress, right? If you imagine it's 3-dimensional, but I'm just trying to keep it, less clutter. That's why I'm only showing you like a slice of it, right? But imagine this is like a 3-dimensional structure, okay? And you will have all these, heads over here. But we're not gonna draw that, like I said, to keep it simple, right? So why don't we put another protein here? Okay, and then we just draw a little bit more phospholipids. There we go. Some proteins are not by themselves. Some of them have sugar attached onto them. Okay, so that makes it a glyco. Glyco means sugar, a glycoprotein. And then some of the lipids might also have sugar on them. That makes it a glycolipid. Just very briefly, the glycolipid and the glycoprotein, it plays a role in cell identity. More on that later. Right, but basically, it allows your immune system to distinguish what belongs to the body and what does not. Gives it cell identity. Now, some of these membrane proteins, they will help attach to cytoskeleton. Okay, so these are little red fibers, those are the cytoskeleton. Right? And the proteins would allow the membrane to be anchored to some of these cytoskeleton. Cytoskeleton could be outside as well, doesn't have to be just on the inside. Okay, so that's a summary of everything that's going on in the cell membrane in terms of the composition and the function of some of the components. Does anyone have any question? No? All right. So let's go back to our study guide right here. And we're gonna look at the function and of the membrane protein. So there are a total of 6 functions associated with these membrane proteins. And some of these proteins are integral protein. It goes all the way through. Some of them are just peripheral protein, but the functions are similar. Okay, so the first one here is to provide attachment to cytoskeleton. Attachment to cytoskeleton. Right? So what does that do? Well, that allows cell to move around in the extracellular matrix. That's like the area outside of the cell. And also, by attaching to cytoskeleton, it helps maintain cell shape. Okay. That's the first function. So you can see here, this is the protein, and all these fibers, all these green things, yellow things, these are the cytoskeleton. Okay, this is the inside of the cell, and out here, that's the extracellular matrix, the ECM. All right, second function here, it is for cell signaling. Right. Let me just draw a diagram for you to help you understand this. So there is your cell, and on the surface of the cell, you would have these receptors. And now these receptors, they would have a particular shape. Okay. And okay, so let's label that first. That these are receptor. And the shape has to be corresponding to the signaling molecule. So in order for the molecule to bind to it, it must have the complementary shape. Okay, you see, so like this pointy end is gonna fit right there. If you have something that looks like a square, right, like that's not gonna fit, right? So it has to be the corresponding shape, like a key and a lock, right? I keep on saying. So these are signaling molecule. And there are different types of signaling molecules in the body, such as hormone. Okay. Now, the hormones don't physically go into the cell in most cases. It will bind to the receptor, and then it will trigger a series of events. And these series of events will change the cell behavior. Change cell behavior. Okay, maybe it will tell the cell to make more glucose. Maybe it will tell the cell to turn on or turn off certain genes. Okay, whatever the case, depending on what the signaling molecule's purpose is, right? The example I gave in class is if someone is getting ready to do a sprint, like running really fast, then you know the signaling molecule, the adrenaline, is going to help the person release more glycogen, right? To break down the glycogen and release more glucose. So that relies on the receptor to respond to the signaling molecule, and the receptor is made up of protein. Okay, so cell signaling, basically we can write the protein acts as a receptor for signaling molecule. I think this is a little bit too thick. Once activated, once the receptor is activated, it creates a series of reactions inside the cell that changes its behavior. Okay. Now, because of this key and lock relationship, even though your cell is exposed to many signaling molecules at any given time, but it will only respond to those that it has a receptor for. Okay, that's that. Next one here, this picture shows enzymatic activity. Now, in the body, a lot of reactions are not going to be finished in one step. So you might start with reactant A, and then enzyme number 1 will change it to reactant B, and then you might have to change it to C by a second enzyme. So it makes sense for your body to put these enzymes right next to each other. So maybe enzyme 1 is here, enzyme 2 is here. Put them next to each other in the membrane, so they're not just like floating around in the cell. And so, you know, triangle here represent A, enzyme 1 will change it to B, and immediately enzyme 2 will take up B and then change it to C. Okay, so this, by putting the enzymes close to each other on the cell membrane, this setup allows chemical reaction to take place rapidly. Okay. And these enzymes, 1 and 2, you can have more, like 3, 4, 5. These enzymes belong to the same chemical pathway. All right. So just want to take a moment to pause here. Does anybody have any question? No? Everyone's good? All right. Let's take a look at the next one. This one shows intercellular junction. Inter means between, so between two cells. Junction. Right? Allows two cells to basically stick together and create a tight seal. So here we have cell number 1. We have cell number 2 here. And then this connection, that creates the intercellular junction. The purpose of having an intercellular junction is to prevent movement of substances between cells, right? Something like this would be particularly useful if you think about the intestine. Right? So there you would have the cell that makes up the walls of the intestine. Okay, and then you would have the food passing by. You don't want the food to kind of leak between the cells, right? You want the food to stay inside your intestinal tract, and to keep it that way, you need the intercellular junction. Same thing for blood vessels. You don't want the fluid to leak out of your blood vessels, so it makes sense to create these tight junctions between the cells. Okay, so prevent movement of substance between cells by essentially linking adjacent cells together. Okay, staples them together, glue them together. All right, next function that we learned in the recording is cell-cell recognition. Okay. In cell-cell recognition, we use, okay, so like immune cells, for the most part, immune cells use glycoprotein and glycolipids as, as identification tag. To distinguish self versus non-self. Okay, so on the surface of all your cells, you have these markers, these glycoproteins, lipids, that gives it an identity. And all your cells will share the same identity. And no one else would share the same thing as you, okay, unless you have like an identical twin. Everybody is unique in terms of the kind of molecules they have on their cell surface, which is why, like, you know, if you think about transplantation, you can't just randomly take someone's kidney and transplant it into anyone else, right? The body is going to reject it because of this cell recognition ability. And so you try to match it up as much as you can. And usually those people would have to take drugs to dampen their immune system so they don't reject the organ. But at the core of it, this ability is important for our immune system, right? Your body has to know what cell to attack and what cell to leave alone. You want to be able to attack the bacteria that are going into your system, but you want to leave your own cells alone. In cases where the immune cells fails to do that, that would cause you to have what's called like autoimmune diseases. Your cell might end up attacking yourself, and that usually is not good. Does anybody know any autoimmune diseases that you might have heard of, read about? Anyone?
Angioedema.
Yeah, I mean there are so many different examples, right? Any other examples?
HIV/AIDS.
Sorry, didn't catch that.
AIDS or HIV.
HIV is not autoimmune, but good try. HIV is more like immunodeficiency. Like in that case, you don't have an immunity. Okay, autoimmune system is when you attack your own self, right? You might have heard of multiple sclerosis. You might have heard of like things like ALS, right? So that's when your immune cells fail to distinguish self versus non-self. Okay, so here, this is a cell Okay, and with some glycoprotein here, and this would be like a white blood cell. Okay, that's checking out the glycoprotein and see whether it belongs to the body or not. In any case, that's something that we'll learn about next semester in the immune system. Last function is transport. Transport. Okay, so a lot of the integral protein plays a role in moving substances across the cell membrane. Okay, so move substances across the cell membrane. Might require ATP. That's energy, right? And the kind of substances that it moves across would be things that are very large. Those don't cross the membrane on their own. They might be charged. Okay, they might be polar. And those things can't cross on their own, and therefore must rely on the help of a membrane protein. So that's the 6 functions associated with membrane proteins. Let's do some poll questions, huh? Well, this one is not poll. This one is just, discussion. And you want to pay attention to this question because it's gonna come up in your lab report, okay? While I have your attention, like there are like 20 of you here. Don't go early, okay? Try to stay until the end because I'm gonna explain what needs to be done for the lab. And you certainly want to stay for that. So how would the percent of saturated, unsaturated fat and cholesterol content of a cell membrane found in plants growing in cold environment differ from those growing in a hot environment? Okay, so saturated fat, as in like the the phospholipid, and then unsaturated and cholesterol. So in the cold environment, what do you expect these things to be like? Is it high? Is it low? No difference. Okay, so I'm gonna give you just a moment to think about it, and then if anyone wants to share, please go ahead. Do you guys understand the question? Like if you take a look at the cell membrane of a plant that grows in a cold environment, what do you expect their saturated and cholesterol level to be? Do you expect them to be higher than a plant that is in a hot environment, lower, no difference? Let's talk about cholesterol first. Okay, what do you guys think? More or less? Why don't we take a vote? Okay. More cholesterol, less cholesterol, or no difference? A is more, B is less, C is no difference. Okay, well, I have about 13 people who've answered. Thank you. And it's kind of half of half. Like some say more, some say less. All right. The thing you have to understand here is in a cold environment, things tend to freeze up, right? Okay, you put water in the freezer, it becomes more solid. So to counteract that freezing action, okay, to counteract the cold temperature, the membrane needs to be more fluid. Okay, so it doesn't freeze up easily. Otherwise, the plant is gonna get damaged, right? And so if you want it to be more fluid, do you need more cholesterol or less cholesterol? Well, earlier I said that the cholesterol, more you have, the more fluid it's gonna be. Right? So the answer is that you need to have more cholesterol. Okay. Similarly, if you want to make the membrane more fluid, do you want more saturated fat or do you want more unsaturated fat? Which one's going to make your membrane more fluid?
Unsaturated.
Unsaturated is going to make it more fluid. That's correct. Right? And you want less of the saturated. Okay. So if you take a look at the membrane composition of a plant, they are likely to be different depending on where the plant's native environment is, right? So if you take something that's from a cold environment, that is what you would expect. And then if you take it from like a harder environment, the opposite is true, right? So each membrane composition will be uniquely adapted to that particular environment. Okay, so remember this when you are doing your lab.
Vincent, please, could you explain this again? I'm a bit confused.
Sure, no problem. Okay. Do you understand this part over here?
Yes, I do.
Okay. So to make the membrane more fluid, you want to adjust the composition of these various components. Okay. Now we learned that saturated fatty acids tends to be solid at room temperature, right? Remember we learned that in Unit 2?
Yeah.
So if you want the membrane to be more fluid in general at a colder temperature, you don't want it to freeze up as easily, you actually want less saturated fat, right?
Okay.
Yeah. So that, you know, when you put it in the fridge, so to speak, it's not gonna freeze up, right? You know what I mean? And that's why you need less of it, and then you will want more unsaturated and cholesterol. Okay.
Okay, I get it now. Thank you.
Good. All right, so that's that. All right, here is a question about fluid mosaic model. It's very lengthy, but it's what we have been discussing so far. So why don't you give it a try, please? If you do not see the poll, you can just type the answer in the chat box. That's another way to participate, okay? Do encourage you to participate because some of these questions are questions that you see on the quiz, okay? Maybe another half a minute or so. Okay, I'm going to go ahead and take it up. We are looking for the true statement. They are free to depart from the membrane and dissolve in the surrounding solution. That is definitely not true. Because if that's the case, then your membrane would just spontaneously degrade and the cell would die, right? So that's not something that happens. And another reason why you know this is not true is because the membrane is made up of the phospholipids. and the surrounding solution is mostly water, right? Mostly water. So things that are lipid and water, they don't mix together. So it's impossible for your membrane to just dissolve in the environment, okay? They occur in an uninterrupted bilayer with membrane proteins restricted to the surface of the membrane. That's not true either, because if you look at our fluid mosaic model, they are not just at the top. They could go all the way through, or they could be at the inside, at the bottom, so to speak, right? It doesn't have to be on one single side. So that is not true. They have hydrophilic tails in the interior of the membrane. That is not true. They are supposed to be hydrophobic tails. Again, you can look at the diagram that we drew, right? In the middle between, that's the hydrophobic tail. It creates a hydrophobic barrier that would block things from moving past it. They can move laterally along in the plane of the membrane, right? That's the correct one. When I was drawing the model, I told you, right? You can, rewind to the recording and listen to the explanation again, right? These phospholipid is free to move side to side, but they sometimes, rarely, could also flip over. But primarily, they just, go back and forth like that. So that makes that the correct one, and this is incorrect because they don't frequently flip-flop, they occasionally flip-flop. Okay, so the correct answer is D. Okay, this one is relatively easier. Hopefully we will have more participation. Go ahead, please. Okay, so that was a minute. And the correct answer is protection from lysis. So of the 6 functions that we talked about, all these are included, but it doesn't protect the cell from bursting. That's the answer that we're looking for. Okay. All right, next question talks about catabolic reaction, and that means we have to fill this thing up before we do it. So here's the next diagram. This picture is actually taken directly from your PowerPoint slides, but we'll fill it in and talk more about it as we go. All right. So there are a bunch of chemical reaction that takes place in our body. Collectively, we call them metabolism. Metabolism. Which again is all the reactions that take place in the body. And we can separate them into 2 categories. We have anabolism or anabolic reaction. This is for building things. So like you might build a protein that you need for cell signaling. You might build protein that's needed for structure. And the other one is catabolism, which is to break things. Breaking things. Okay. And the 2 reactions are actually related to each other, as we will see in the diagram below. So over here, we have macromolecules from our food. Macromolecules from food that we eat. So there might be some proteins, from the meat, from the cheese, from the beans. There will be some lipids, some carbohydrates, and even some nucleic acid. Now, we cannot use these macromolecules as they are, and we must break it down first. So to break down the macromolecules, is it catabolism or anabolism? Which one involves breaking macromolecules?
Catabolism.
Metabolism.
That's right. So kata is same prefix as like catastrophe, right? In catastrophe, everything breaks down. In catabolism, we are breaking the food down. So catabolic reaction or catabolism, both is okay. So in breaking down the food, we extract the energy from the food and we temporarily store it in the form of ATP, adenosine triphosphate. So this is the preferred form of energy in cells. It's the currency that your cells use to do work. And as a byproduct, we generate heat. So heat is good for warming up the body, right? Especially on a cold day like this, after a meal, you feel warmer. That's because when you break down the food, it generates heat to keep us at 37 degrees Celsius. And at the end of it, you are gonna end up getting many building blocks for biosynthesis. Biosynthesis just means to build things, okay? And these are monomers, right, from your food. And the monomers. An example of monomers would include things like amino acid, maybe a little bit of glucose, maybe some fatty acids, right? So all these things are now available for you to use to make molecules that you need. And that's what your cell does. Right? Your cell is going to take all these building blocks and will link them together in anabolism or anabolic reactions. Right? So the amino acids will connect together to form proteins. Your ribosome does that for you. The fatty acids might come together to form various type of lipids. Your smooth ER does that for you. Right? So that requires ATP, that requires energy. When you build things, it requires energy. And at the end of it, you end up getting macromolecules that are needed in the body. Okay. So the key thing here to know is that the energy, ATP, right, released from catabolic reactions, from breaking down your food, is used to drive anabolic reaction. Okay. This requires energy, and that energy is supplied by the catabolic reactions, which kind of makes sense, right? Like we got to continuously to eat in order to survive, to live, right? And that's because it creates these ATPs that would supply our body for all the necessary reactions that keeps us alive. Okay. Any questions about this? Please ask if you have. No questions? Okay, well, I have a question for you. Let's try this out. This is a multi-select question with more than one correct answer. Please choose them accordingly. Okay, thank you. Catabolic reaction, that is for breaking things down, right? So A, consume more energy than they produce. It's actually the other way around. If you look at what's written here, right? They actually release energy to produce ATP, right? So you must release more than you capture. That makes this the incorrect choice. Breaks down polymers into monomers. That is correct, right? Breaks down the food that we eat. Produce excess heat to warm the body. That is also correct. And it occurs in living organisms in general, not just human cells. Okay. So B and C. Okay. Moving on. Now, what do we use ATP for? So just to quickly recap for you, in case you need a refresher, this is the ATP molecule, and the ATP, when you add water to it, is going to separate into ADP and a phosphate and a P, and in the process, it's going to release energy. And what you want to do is you want to recharge the ADP so that you can form ATP again. So you're going to supply energy. And as I just showed you, this energy comes from catabolic reaction, right? Catabolism. Okay, so that's gonna create energy, and the energy is used to charge up the ADP and to create ATP again. And when you need to use the ATP, you add water and you break it down. And then what is this energy used for? Well, this energy can be used for anabolism, like we explained above, but it can be used for other kinds of work as well. So in the lecture, we talk about how it can be used for mechanical work. Mechanical work. So that would be like some kind of physical movement. Okay, so you know, to move your hands, to move your legs, to walk around, to run around, all that physical movement actually do require ATP. Right? So example, muscle. Right? That's called mechanical work. Chemical work. So such as anabolism, enzymatic work. Those requires energy. And the last type of work is transport work. If you watch the lecture recording, you will know that there are two types of transport. Those that requires energy, we call that active transport. Okay? More on that later. All right, next we have factors that affects the rate of diffusion. Just to give you the definition of diffusion again. This is something that happens spontaneously. That means on its own without any energy requirement. And it involves substances. Substances will move from area of high concentration to an area of low concentration. Think about opening a bottle of perfume in the corner of a room. Eventually, the whole room is going to fill with that scent. Doesn't have to be perfume like, those Febreze thing, right? People spray, the whole room is going to be filled with that scent. And that's an example of diffusion. So there are factors that affects the rate of diffusion. Does anybody remember anything from the lecture? Any of the 4 factors? Okay, Manish says temperature. That's correct. Okay, temperature. That is absolutely correct. Right, so higher temperature equals faster diffusion. Okay, like you will be able to smell the garbage easier on a hot summer day than like a cold day like today, right? Because the diffusion is slower on cold days. Yes, pressure is another one, but that's not one that we talked about in class, so I'm gonna leave that out. Anyone else? Size, that's right. Ping Xia said size. Bigger molecules takes longer to move around, so that would be slower diffusion. 2 more. Anyone else? Size. Tamara said charge, and then let's write that down first. Charge is correct. Okay, so this one is a little bit harder to write out exactly what's going on, but I will use an illustration here for you. Okay, let's say there's a membrane, and then we have a bunch of H plus here. Okay, positive charge. So like charges repel each other, right? Like positive repels positive, negative repels negative, right? So if you have a bunch of positive here, initially you will push them over, and it will cause it to try to move away from each other very quickly. And that repulsion actually speed up the process initially. However, as the process goes on, as you have build up more positive on this side, then these positive charges will kind of push back, right? Because of the light charge repulsion. So later on, it would kind of slow the process down. Right? So it depends on which phase of a diffusion you're talking about. It would have different effect. One last one. Yeah, I mean, like it depends on the type of membrane you have as well. But again, that's not something that we have in the notes. Yeah, the steepness. That's right. Steepness of the gradient. Gradient is just a fancy way of saying difference. Okay, so how much of a difference are we talking about, right? The greater the difference between 2 points, the faster the diffusion is going to be. So let's say again we have 2 chambers here separated by a membrane. Right? Let's say we have a 70% sugar solution versus a 10% sugar solution, right? So the difference here is 60. That would cause diffusion to happen quite quickly. On the other hand, if the difference is like, you have 70% solution here and 60% solution here, it's only a 10% difference. You will still have diffusion, but it's going to be much more subtle. Okay, so this is faster diffusion and slower. The greater the difference between 2 points, the faster the diffusion is going to happen. Okay. Now there is something that I talked about in the lecture, and it's probably a good idea to go through it again. You will see a question like this either on the quiz or on the test, and it's probably a good idea that you try to understand it now and ask questions if it doesn't make sense. So here is the tube, U-shaped tube, and it's separated by a membrane here. So this membrane is special. It only allows water to pass through, nothing else. That's a key thing to know. And this tube is gonna have equal water level on both sides initially. Just gonna shade it in. There we go. Okay. So nothing out of the ordinary. It's pretty much what you expect. The water level would be the same. And then this is what happened. Okay, we're gonna dissolve some solute into the solution. And then I'm gonna put more here than on the right side. So here is my question. What is going to happen to the water level here? Okay. Is it going to go up? Water level. Okay, this is gonna get more water. Is it gonna go down? Or is it going to be no change? Okay. In other words, which way will the water move? Will the water move this way? Will the water move this to the right? If the water moves to the left, it will cause the water level to go up here, right? If it goes to the right, it will cause the water level to go down, right? So that's my question for you. And please choose something from the poll. Or type something in the chat. Okay, your participation is important because it tells me whether the class understands or not. I'm just gonna wait a little longer, maybe another half a minute, just for people to make up their minds. It's okay if you get it wrong. Just need to know what people are thinking. Thank you for writing the answer in the chat box, Chico. Repeat the question. Okay, the question is, which way will the water go? Right? If the water goes to the left, it will cause the water level to go up. If the water goes to the right, it will cause the water level to go down on the left side. Or if the water doesn't go anywhere, then there will be no change, right? Okay, well, let's take a look. We have 50-50 here, almost 50-50, right? And we will see what the correct answer is. Okay, so pay attention. I guarantee you will see this question either on the quiz or on the test later on. Okay, so make sure you understand this. All right. So the membrane at the bottom only allows water to pass through, right? So these little red balls, they're not going anywhere. Okay, I'm not asking you where the red balls will go, right? I'm asking you where the water will go. So because the red solute is not able to move, the only thing that will move is the water. And water will try to balance out the concentration on both sides. So if the concentration on the left side is higher than the right side, it will suck the water towards that end. Okay, so water is actually going to move in this direction from left to right. From right to left. The water will move left and it will cause the water level to increase. Okay. And then subsequently, the water level here will go down. Do you understand that? Okay. So let me write it down for you first and then I'll explain more. Water moves towards the more concentrated side to dilute it out. Okay, think about it. As the water moves to the left, you will dilute the left side and you will make the right side more concentrated, right? And the water will start moving when the concentration on both sides are the same. Okay. Another way to think about this is like if you put a spoon of salt into your mouth, what's gonna happen? It's gonna suck the saliva out of your mouth, right? And that's exactly what this is doing. You have more salt, more red stuff on the left side. It's sucking the water towards itself, right? To try to balance out the concentration. Okay, so the correct answer is the water on the left side will go up. Oh boy, what did I press? The water on the left side will go up. Okay, water level on the left, okay, goes up. Please ask a question if you need clarification. Okay, so this concept is called osmosis. Okay, osmosis.
Please, can
Yeah.
you go over again? It really baffles me.
Okay, sure.
I'm
Sure.
confused.
Yeah, no problem. Okay, so let's just try this one more time. So again, right, like you have the tube, and then the water level was the same initially. And here's the membrane, and the key is that it only allows water to pass through. Now, if we put more solute on one side than the other, okay, I know what a lot of you are thinking. A lot of you thinking, well, things goes from high concentration to low concentration, right? So wouldn't it make sense for the red balls to go to the right? Okay. Now that would be correct if the membrane actually lets them go through, right? But it's important to know that in this particular case, this is only permeable to water. Okay, so the red dots cannot move. And so having higher concentration on the left side is going to suck water towards the left side in an attempt to balance out the concentration, right? So what happens if you add more water to the left side? This gets diluted, right? It's like, you know, drinking juice. If it's too sweet, you can add some water, it dilutes it, right? On the other hand, if the water is leaving here, it gets more concentrated. It's more concentrated. Okay, so over time, as the water moves to the left, the water level will rise on the left, and then it will decrease on the right. And eventually, you will dilute the left side enough, and you will concentrate the right side enough for the two sides to be the same. Okay, and that's why the water level is going to go up on the left, and it's going to come down on the right. Any follow-up questions? If this is a difficult thing for you to visualize, again, we just put some salt in your mouth, right? Like just a little bit, then immediately you will feel all the saliva being pulled out by the salt, right? This is no different from that. Is that okay? Was it Francisca, I think, who asked the question?
Yeah, I'm okay.
All right.
Thank you.
No problem. Okay, let's go back to this thing here. I think we're gonna skip the break here, okay? Because we still have a little bit to go through, and then we have to talk about the lab. Next, describe what happens to a cell in each of the following solutions. All right, so we have three things going on here. We're placing an animal cell in a solution, and the solution is either isotonic, hypertonic, or hypotonic. So the first case here is isotonic, and if you cannot see the dots clearly, maybe I can emphasize that for you. In this case, the concentration of the solute, the black dots, right, which was the red dot previously in the U-shaped tube.
Please, can I ask this before you proceed?
Sure.
In your previous recording, the one you asked us to watch, the factors of the rate of diffusion, you made mention of the charge. I really don't understand. Yeah, the charge.
Okay, so the charge, right? Okay, the charge over
Yeah.
here. Do you understand light charges repel each other?
Yeah.
Okay, so like if you have a bunch of positive charges over here, they will push each other away, yes?
Okay.
Right, because they are the light charges, right? And they don't like to be close to each other, so they will push each other away, and that pushing force will speed up diffusion at the beginning. Is that part okay?
Okay, thank you.
Yeah, it's like having a magnet, right? It's gonna repel them, right? But eventually, when you have enough positive going to the other side, they will try to push back. And so it will slow down the diffusion towards the end. Okay.
Okay.
So think about that. And if you still have questions, feel free to email me and we can talk about it. Okay, so here is isotonic, which means the concentration can be represented by a square bracket. So the concentration of solute on the inside of the cell is equal to the concentration of the solute in the solution on the outside. So in this case, the rate at which water enters the cell, represented here by the red arrows, will be equal to the rate at which the water is leaving the cell. And you have the same amount coming in as it is leaving. So rate of water coming in equals to the rate of water going out. Okay, and in this case, there is no change, no net water movement. Okay, no change in cell shape. One way to visualize this is think of a bucket of water. Okay, so we have a bucket of water, fill it up, and there is a tap. There is a tap. Okay, we're going to turn on the tap, so the water is going to be coming in like that. And our bucket here is going to have a hole. And so the water is going to be leaving at the same time. So if the rate at which the water is entering the bucket is equal to the rate at which it is leaking out of the bucket, then the water level does not change, right? So same situation here. The cell is not getting bigger, is not getting smaller. There's no net change in the cell shape. If we are talking about an animal cell, they are going to be happy. Okay? This is the preferred state for animal cells. Your blood should be isotonic so that your red cells are going to be happy. If we are talking about a plant cell, I mean, they are okay, but not the best. Okay? It's not the preferred state. Right? They will survive, but they will not necessarily thrive. Okay. So that's isotonic. Same concentration inside and outside. The next one shows a hypotonic solution where there are higher solute on the inside compared to outside. Okay, more black dots on the inside, right? So concentration of solute on the inside is greater than concentration of solute on the outside. And in this case, you would have more water coming in. Remember, the water always goes towards the side that has higher solute concentration, right? So you would have more water coming in than leaving. So rate in is greater than rate out. Okay, so net water movement into the cell. What that means is you have more water coming in than leaving the cell. So what's going to happen to the cell volume? Well, cell volume increases, right? So in our water tap analogy, you would be turning on the tap really, really big, okay, when the water is leaving. But because more water is coming in than leaving the bucket, your water level will rise over time. And eventually it will overflow, right? So in the case of an animal cell, this is bad news, okay? It will burst. We call that lysis. But plant cell, they like this. They are happy, okay? The plant cell will not burst because it has a cell wall, right? So won't burst because of cell wall. Okay. So in this case, you have more water coming in than water leaving. Are we together so far? Any questions about these two things? Okay, so this is hypotonic. Yeah, sure,
Did
go
you
ahead.
say the hypertonic solution, the amount of water in the cell is equal to the amount of water outside the cell?
For the hypotonic or the
No,
isotonic?
the hypertonic, yeah.
This one?
The first one.
The first one is isotonic. Iso.
Oh,
Yeah, isotonic. Okay, then the solute is the same on the inside as it is outside. Okay.
okay.
Yeah, this one is hypotonic. You can remember the big O. That tells you that the cell is swelling up. Okay.
Okay.
Anyone else have any other questions, comment before we move on to the last case? Okay, the last case is hypertonic. Okay, and in this case, you have more solute on the outside than the inside. And so you have water coming in, but you have more leaving. More leaving. So solute on the inside is less than solute on the outside. Rate in is less than rate out. And then you would have net water movement out of the cell. And the cell shrinks. Animal cells, they're okay. Again, not the preferred state, but they will live. This is what happens when you don't drink enough water, you dehydrate it. Okay, so this is called plasmolysis. And if it's a plant cell, they are not happy. Right? If you forget to water your plants for an extensive period of time, they become like wither, right? Shrivel up. Hypertonic. For the water tap situation, right? It's like having a really big leak. Okay, so like it's pouring out, and then you have your normal tap. Because you're leaking more than the water is coming in, over time, water level is going to go down, right? Okay, so those are the three cases. I hope that makes sense to you. Next, we're going to move on to this active transport and passive transport. So we have the active transport here. And in the case of active transport, the movement of molecules is going to go from low concentration to high concentration. Okay, square bracket means concentration, huh? And this goes against concentration gradient. It's like going uphill. That's why it requires ATP. From the cell. We learned about several examples of active transport in the lecture recording. There is bulk transport, as well as protein pump, such as the sodium potassium pump, right? For passive transport, it is the other way around. It goes from an area of high concentration to an area of low concentration. And this is like flowing downhill. That would be down concentration gradient. And because it's going downhill, this one does not require ATP from the cell. Something else is powering it, okay? Does not require ATP. Example of passive transport, we have osmosis. We have facilitated diffusion. Okay. So that's comparing the two. Okay, we can do a quick summary of all these things. We have transport, right? Transport, let me choose a thicker pen here. Transport. And transport is separated into active and passive transport. You know what? Let's switch them because the active one needs more space. Okay. So for passive transport, we have things like simple diffusion. So simple diffusion is best for small things, small non-polar. If it's a hydrophobic, they will be able to cross the cell membrane just on their own. Okay, no help required. So example would be something like, O2, right? CO2. These things can just cross on their own, no problem. Passive, another example of passive transport would be osmosis. Okay, so this is the diffusion of water, basically. So the water will move in or out depending on whether the solution is hypertonic or hypotonic. And that is passive, doesn't require ATP. And what's the third type of passive transport? Does anybody remember from the lecture?
Facilitated.
Yeah, exactly. Facilitated diffusion.
Okay.
Facilitated diffusion. Okay, so this is moving things across the cell membrane with the help of a membrane protein. Membrane protein. So that would be for things that are like large, maybe charged, okay, or polar. They don't cross the membrane very well on their own, and they must require the help of a membrane protein. So like glucose, right, that's one example. Okay, some electrolytes, ions, these things don't cross on their own. So there are two types of facilitated diffusion. One of them is called the channel protein. And then the other is called the carrier protein. Okay, so here is your phospholipid bilayer. The channel protein is an integral protein and is exactly what it sounds like. It creates a channel or a tunnel in the middle. And whatever you're trying to move across, right, they will just go right through. Okay, they're just going to go through. So that also requires no energy, just creating a tunnel that shields the molecule from the hydrophobic tails, and it's going to be able to pass through. The other one is carrier protein. In this case, the carrier protein is like a revolving door. Okay. It's like Pac-Man. It will grab on to the substance, and then it will rotate, and then it will split it out on the other side. Like that. Okay. So in the case of channel protein, it's just a tunnel. The protein itself does not change shape. But for the carrier protein, it physically grabs onto whatever you try to move, and then it changes shape and then split it out on the other end. Okay, for active transport, we have protein pumps. Protein pump. And the only example I gave you is the sodium potassium pump. Okay, you can read more about it in the lecture notes. And then the other one is bulk transport. Bulk transport. So bulk transport comes in two forms. We have exocytosis. That's moving things out of the cell in bulk. Maybe you need to release some proteins that you made. Right? You want to release them from the vesicle. You can do exocytosis. On the other hand, we have endocytosis. And depending on what you're bringing in, you will have different forms of endocytosis. There's phagocytosis, which is bringing in solid. So this is cell eating. You have pinocytosis, which is bringing in liquid. So that is cell drinking. And then the last one is called receptor mediated endocytosis. It's same as the other two types, but it requires the molecules to be a particular shape before you can bring them in. Okay, so there's just an extra layer of control and regulation there. Okay, so this summarizes all the different types of transport. And again, for the full story, for all the details, you can watch the recordings on that. Okay, let's go back to here. Choose the more concentrated solution in each of the pair. Okay, we have 80% water and 20% starch versus 90% water and 10% starch. Okay, so this one is quite obvious. The one that has more starch is going to be more concentrated. Next one requires you to do a little bit of math. You have 5 grams of salt, NaCl, in 50 grams of water versus 5 grams of salt in 25 grams of water. So I can take this and then do some calculation. 5 grams of salt in 25 grams of water. Now I can try to make the water the same amount as the other one so that we can compare it. So I will multiply this by 2. But the rules about fraction is if you multiply the denominator by 2, we must also multiply the top by 2. So that will give me 50 grams of water, and that would be 10 grams of NaCl. So now if we compare 10 grams of salt in 50 grams of water versus 5 grams of salt in 50 grams of water, we will see that this is the more concentrated one. Last, we have 85% solvent, that's the liquid portion, and 15% solute, 75% solvent, and 25% solute. And once again, the one that has higher percentage of solute, that's the more concentrated. Okay. Number 9, list 3 types of bulk transport. We just did it on the diagram, so I'm not going to repeat it. It was the phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, so all these long answer questions, I will post the answer online as well as the fill in the blanks, so you can check your answers against mine. I want to spend the last little bit of time going through the lab with you. Please pay very close attention about the lab here. It's actually very easy, but people don't do well because they don't follow the instructions. Okay, so just try to listen carefully to what I tell you to do, and follow the instructions. That's very important. Okay, so here is the course homepage, and I will post a link to the lab later on. Right? But if you try to find it, you're on your own. You will click on content, and then click on virtual labs. There are 2 labs. We're doing the beetroot lab next week, so click on that. And this is the beetroot lab. The purpose of this virtual lab is to investigate how temperature affects the permeability, the membrane permeability of beetroot, okay, Beta vulgaris. Before completing this lab, please review the lecture on cell membrane structure and transport, which is the lecture we just did, right? You should also review the first lecture, the scientific method, okay? Specifically, you should understand the independent variable, dependent variable, as well as standardized variable. All that stuff will be important when it comes to doing your lab report. What you need to do is, because you are not physically doing the lab, you will watch a video of the lab that is being done, and that's this YouTube video right here. Okay, so you're gonna click on it, and then the person doing the demo is gonna explain a lot of stuff. Okay, it's only 8 minutes. And I can tell you from experience, many people try to either skip through the video and try to find answers to do the lab, or they just completely skip the video and do the lab. I don't know where they find the answer. Okay, maybe they try to find it on the internet. Maybe they try to just make up answers. And those are the people who do very poorly in the lab. A lot of the questions that are being asked in the lab report is actually related to what is being said in the video. So just watch the thing. It's only 8 minutes. Okay, take some notes and then watch it again if you need to, because that's the only way to see what the experiment is about and what kind of results they're going to get. Okay, please watch it. And I can easily see who has watched it and who hasn't. Okay, like that. I'm able to track that kind of information. So, if you didn't do well in the lab and you come back and email me and say, oh, can you give me another chance to submit the lab? The answer is probably no. Okay, like you have to do it right the first time. You have to watch the lab demo here. Once you've watched it, you have to download this document. Okay, so you're going to click on that, and you will see the thing here, but you cannot fill this in. Right? So you must download it. There's a download button at the bottom. So you click on that, and it's going to go onto your computer. Okay, so I'm just gonna open it. You might have to click enable editing. Okay, if you don't have Microsoft Word, you can use the computers at the school, and I think you actually can download a free copy of Microsoft Word to use online. Okay. Oh, this is very important. Anybody uses a Mac computer here?
I use that Mac computer.
Okay, yeah, thank you. Okay, and other people as well. If you use a Mac computer, you have to export your report as like PDF, okay, before you submit it. Because I have PC and there is no way for me to open the Mac format, right? It's like .page or something, right? So I won't be able to grade your assignment if it's not in PDF or in Word form. All right, so please export the document. Thank you. If you have trouble doing that, you can email me, and you know, we can sort it out. Okay, I'm sure there are some videos online that shows you how to do that. Okay. All right. So the lab is just answering these questions. So for example, first part here, 2 marks. What is the purpose of this experiment? Well, the lab over here, if I just go back, right, it says the purpose of this experiment is to investigate how temperature affects membrane permeability of beetroot. You can just copy that. Okay, so you will literally just type your answer here. The purpose of this lab, of this experiment, is to investigate the effects of temperature on membrane permeability of beetroot. There, that's 2 marks, okay? You can type that exact same thing in your lab. Okay? I give you permission to copy that. Okay? That's 2 marks. Okay? That's all I'm asking. You don't have to go on, you know, internet and try to find answers. And I will know if you copy the answers from the internet. Okay, I've been doing this for a very long time. So if I see an answer that seems out of place, and I'll be able to search it up. Don't do that. Just answer it on your own, based on the video, based on what we learned in class. Write a hypothesis for this experiment. Many people lose marks here because of 2 reasons. Number 1, a hypothesis is not a question, it's a statement. So when you write a hypothesis, make sure it is a statement. Don't give another question. Don't say, will the temperature affect the membrane permeability? That's not the hypothesis. That's asking a question, right? Secondly, people will also lose mark if they say something like this: the temperature will affect the membrane permeability, period. Okay, that's a bad hypothesis because you did not predict how it will affect the membrane permeability, right? Do you think the temperature will increase membrane permeability? Do you think it will decrease membrane permeability? Do you think it has no effect on membrane permeability? You have to write it out explicitly, okay? So it's okay if your hypothesis is wrong, but you have to pick a side. You have to let me know what you think. Does that make sense? Okay, so that's 2 marks. Next, state the dependent and the independent variable. So watch the experiment, the demo, see what was being changed and what is being observed, right? If the purpose is to investigate the effects of temperature on membrane permeability, either the temperature is independent variable or the membrane permeability is the independent variable, right? So you have to see which one is being changed and which one is being observed, and then you would answer the question here. Next one, okay? Why is beetroot chosen for this experiment? Okay, why not choose carrot? Why not choose tomato? Okay, so you have to answer this part as well, right? Don't just tell me beetroot was chosen because, right? You have to also say carrot is not suitable for this experiment because, and then tomato is not suitable for this experiment because. Okay. Safety precaution. This is pretty self-explanatory. You know, obviously when you do experiments like this, you have to wear safety goggles, maybe, right? You have to watch out for sharps, you know, standard things. Just watch the demo and think about it. All right, next question here. The test tubes were labeled 20 degrees, 40 degrees, 80 degrees, 60 degrees, and 80 degrees. Which tube is considered as the control of the experiment? Explain what is the significance of having a control. A lot of people lose mark here because they picked the wrong control. Okay, the control is one of these tubes. Okay, so don't make up anything else. Your answer should either be 20, 40, 60, or 80. Okay, so you tell me the control of this experiment is the whatever degree tube. And then people forget to answer the second part. Why is it necessary to have a control in the experiment? So we talked about this in lecture 1, right? Make sure you go back and find the answer. All these are pretty self-explanatory. Limit your answers to 3 or 4 sentences, right? Don't write me a novel. Okay, just keep it short and concise. This is another one. Okay, why is it important to leave the beetroot in the test tube submerged in respective water bath for 20 minutes? And so you will see in the video, the lady said we'll dip them in the water bath for 20 minutes, and then you will put them beetroot in. So why not longer? Why not do it for 30 minutes? Why not do it for like 10 minutes? Right? You have to explore both sides in order to get full marks. I think this one is another one. An example of qualitative analysis. A lot of people just Google qualitative analysis and then they paste the answer here. But for some reason, like if you Google qualitative analysis, the answer that Google gives you is really weird and it totally does not apply to what we are talking about here. Okay, remember in lecture 1, we specifically discussed the difference between qualitative and quantitative observations, right? So that's what this is about. Right? When you're talking about qualitative observation, qualitative analysis, what aspect are you talking about? Are you talking about numbers? Are you talking about using your senses? That's what I'm asking. Okay, and that's what you should look at. Okay, and then they ask you for quantitative, right? So you must know how they are done differently. Okay, over here, describe the relationship between absorbance measured by spectrophotometer and color intensity of the solution. In the video, they actually don't use the word spectrophotometer. They use the word calorimeter. Okay, calorimeter. It means the exact same thing. Okay, so don't let the difference cause confusion. Now, I am going to briefly explain this situation to you. So hopefully it will make more sense when you watch the lab. Okay. So in the lab, right, we're trying to see how temperature affects the membrane permeability. And we choose beetroot because of its bright color, right? It's easier to see the color. Generally, the color stays within the bead, and it's only when the membrane starts to break down that you will see the color, the pigments leaking out. Okay, so you can assess the color using one of 2 ways. You can use your senses, and you can look at the intensity of the color, how red it is. Okay, so that's one way of looking at it. The other way of looking at this is to actually measure how red something is using the calorimeter, okay, or the spectrophotometer. So how that works is as follow. Okay. If I have something that's red, okay, consider some like a block or something that's like a red block. Okay, over here, like we have a red block. Okay, and then this is your eye. Okay. Why is this thing appear to be red to you? When you look at it, why does it look red? That's my question. What kind of light is bouncing off this red cube? You know, like light, right? Light is white light, like the rainbow colors, right? It has like red, yellow, orange, green, blue, whatever, right? So like what color is being reflected by this red object so that it appears to be red to us? Does anybody know? Do you guys understand my question? So the light's gonna strike this block, and then some of the light is gonna get reflected and enters our eye. Which color is being reflected? Really? Nobody knows? A little bit surprised there. Okay. That's okay if you don't know. I'm gonna tell you and you remember it, okay? Red light is going to bounce off. That's why it's red, okay? Every other color will be absorbed, okay? The blue color will be absorbed, the green color will be absorbed, and everything else except for red, that is being bounced off. So the reason we see things as different color is because that particular light is being reflected. Okay, so like this folder, which you cannot see on my on my virtual background, but this folder is blue, okay, because it's reflecting blue light. Okay, the leaves are green because it is reflecting green light. Okay, so whatever color something is, that's the color that is being reflected. Okay, so in our beetroot experiment, instead of just looking at the color and comparing how dark the red is, you can use the colorimeter to check it. Okay, so this is the solution. This is a test tube, and you know it's going to be a little bit red because of the beetroot juice. Okay. And what the colorimeter does is it actually shines green light. Green light. Green light at the solution. Okay. So depending on how red the solution is, the amount of green light that is being absorbed is going to be different, right? Do you understand that? Depending on how much red is in the solution, the amount of green light absorbed will be different. So there is a detector here. There's a detector at the end that would detect how much green light passes through. Okay, this is called absorbance. Okay, how much green light was absorbed? That's the question. So a solution that is more red, do you expect higher absorbance or lower absorbance?
Lower.
Okay, let's do a vote. Okay, please participate. I need to know that you understand this, otherwise the lab's gonna be disastrous, right? If it's darker red, more red, do you expect the absorbance of green light to be higher or lower? A for higher, B for lower. Okay, I have 11 response. I'm waiting for 9 more, 8 more now. Just choose something. Okay, the correct answer is higher absorbance. Okay, because remember, red absorbs everything except for red, right? So the more red something is, the more other colors is going to absorb. If you're using green light, right? If it's really, really red, then basically very little red, very little green will get through. Much of the green is being absorbed. Okay, so higher absorbance means more red. So when would you have more red? When will the solution be more red? When the membrane is very intact, or when the membrane is falling apart, in the case of the beetroot. Okay, when are you going to get more red out of the cell? When the membrane is intact or when it's falling apart? What do you guys think?
Intact.
Intact? Intact means like together, right? Like intact means like there's no damage, right?
Yes.
Okay. Anyone else?
I think when it's falling apart.
Yeah, like think about that. This is the cell, right? This is the cell.
Yeah.
And then you're gonna have all the red pigments inside. Okay. Now, if the membrane is intact, it's not going to be coming out, right? But if the membrane is starting to fall apart, it's going to have all these like holes in it, right? Then the pigment will leak out, and then the solution will become red, right? Okay. So the more the membrane falls apart, the more red, the redder, I don't know what's the right way of saying it, the more red the solution will be, and the higher the absorbance is going to be. Do you understand those relationships? Okay, so keep that in mind as you watch the demo and ultimately when you do your lab. Okay, so here it says, right, describe the relationship between the absorbance and the color intensity of the solution. I basically just told you the relationship. You just have to put that in your own words and write it down, okay? All right, so here are some data. And we have temperature on one side and we have absorbance on the other side, okay? Watch the video. The person will show you a graph and the person will show you the type of graph to use as well as what goes on the y-axis and what goes on the x-axis. All you have to do is create the same graph, okay, using these data. If you have Excel or if you have Google Sheets, you can create it easily. I understand that you guys have learned how to create graphs in your computer class already, so this shouldn't be too difficult. But if you need to refresh your memory, you can watch this YouTube link. Now, this is a 4-mark question. And people lose marks on silly things like they forgot to put a title, they forgot to label their axis, or they use the wrong kind of graph, okay? Watch the video, you will know what kind of graph to use. There should be just one graph, okay? I don't want to see multiple bars, lines, whatever, right? Just one single graph that illustrates this relationship, and then you will paste it over here into the space right here, okay? Okay, so data analysis. At what temperature does the absorbance peak? So you look at the graph and then you see when is it gonna, when does the graph stop rising? When does the graph stop changing? That's what I'm asking for. And do you expect the temperature where absorbance peak would be same for membranes of an other plant? Okay, so think about what we said earlier today, right? At the beginning about the membrane composition, okay? Like from cold environment, hot environment. Think about that when you are answering this question. All right, I know we are running out of time, but I just want to make sure going through this before we end. Provide 2 reasons that explain how temperature affects membrane permeability. Again, the answer for this one is right in the video, okay? And there are specifically 2 stages in which the temperature affects the membrane permeability differently. Okay, to get 4 marks, you need to tell me both of those things. Number 3, this is very easy. What's the conclusion, right? Did temperature affect the membrane permeability, right? Did you get it right? Okay, some people lose marks because they forgot what they wrote in their hypothesis, and then they accept their wrong hypothesis or they reject their correct hypothesis. So just make sure you get your story straight, okay? Whatever you said in the hypothesis, either it was the same as the result, which you will accept it, or it was different, then you will reject it, okay? Now, this one is a little bit of an application thinking question. A piece of frozen beetroot is placed into a test tube of tap water at room temperature, okay? The absorbance recorded 20 minutes, after 20 minutes is 84. Provide an explanation for this observation. Now, so this wouldn't really make sense until you do the graph, okay? Then you would have an idea of what 84 means. Like, is that high? Is that low? How does it compare? Okay, so the hint I'm going to give you about this is to think about what happens when you freeze water. Okay? You might have this experience. You want to have a cold soda drink, cold Coke or something, but you only have one that is sitting in the room. So to cool it down quickly, you put it into the freezer, but then you forgot about it. Okay, and then what happens to it afterwards? Right? Think about that. Or you could just fill up water, like a plastic water bottle, all the way to the top, close it, put it in the freezer. and then afterwards what happens. Okay, so think along those lines, and then you can answer question number 4. So I hope this helps you prepare you a little bit about how to do this assignment. You don't have to give me title page or anything like that. All you need to do is just directly type the answer in the box, and then you submit it. Did you guys learn how to submit assignments online in your other courses yet? Is that something I need to go through? Do you guys know how to do it? Submit assignment online.
We've submitted assignments.
Yeah,
Okay.
I've submitted some already.
Thank you. All right. So it's the same process. I'll put a link there. But for some reason, if you don't know how to do it, you can just email me, and then we'll figure it out. And don't be afraid to email me if you need clarification on the lab. Okay, I want you guys to do well on this lab. It's just easy 10%. It will boost your grades. So if you need help, let me know. I cannot check over your lab though. Okay, so don't send me your lab and ask me to read over it. But if you need me to clarify some of the questions, I can do that for you. So no class next week. The entire next week is for you to work on your lab, and you will hand it in on let me see, on Wednesday, I believe. Okay, so you will work on the lab on the Monday's class, and then you will submit it by the end of the day on Wednesday. Okay, is there time limit for the lab test? No, it's a document, right? So you download it and then you fill it in. Okay, but you have to submit it online by the end of next Wednesday. Okay, so our next class together will be on February 15th. Now you do have another quiz coming up though before we meet again, and that will be on the 13th. That is on lecture 3 and lecture 4. Okay, so I'll post more information about that later on. But first thing first, you should focus on doing your lab. All right. Okay. Thanks again. I'll see you all on the 15th. Okay. Bye for now.
Sir,
Yes.
we will have to prepare your today's lecture and the